and welcome to Scilights, the science highlight show which gives you a breakdown of some of the coolest research coming out this week. My name is Julia Ravey. I am a final year PhD student who should be a doctor of neuroscience in the next year. With coronavirus, we've got extensions, so I'm not quite sure when I will graduate and submit my thesis yet, but hopefully it should be in the next 365 days. Let's crack on with some science in the news. My first news story this week is that a drug in the US has been given approval to be given to coronavirus patients. This is a drug called remdesivir, hope I'm saying that right, and it was originally made to combat Ebola. Its approval came from the results of a small clinical trial, and this trial shows that the drug shortens recovery time of patients with severe COVID-19. So it's not a cure, but it is a step in the right direction. What a shorter recovery means is that patients are spending less time in intensive care units. So that means there will be more free beds for other people who are severely ill. This trial was only on 1,063 people, so it was really short, and they didn't test the drug on mild cases of COVID-19, so we're not too sure how it would work on those people. But those who were severely affected and given remdesivir, they were in hospital for 4 days less, which is a decrease of 31% of disease duration than people who weren't given this drug. In order to get a closer look at exactly how this drug interacts with SARS-CoV-2, a group of scientists in China used a technique called cryo-electron microscopy, and this allows us to zoom in really close to look at this interaction. They found that the drug inserts itself into the new copies of the virus being made. And once the drug is inserted, the next part of the viral sequence can't be added on. This is because remdesivir does not have the right structure for the next part of the sequence to bind. You can think of this as putting a wrong piece of a jigsaw puzzle in place and then trying to get the neighbouring piece to fit. It just won't work. So what you end up with here is a shortened version of the SARS-CoV-2 sequence which the virus needs to replicate. And this shortened version cannot go on to make new viral particles. So why doesn't this drug completely cure COVID-19? So once the molecule has bound into this jigsaw puzzle sequence, it is stuck there. This means that each drug molecule can only block one replication event. And viruses copy themselves a lot more than that. You would need a lot of this drug in the cell to be able to completely stop all the viral particles in there replicating. Early drug trials like this are very hard to interpret, and we must move with caution. A small study in China looking at remdesivir with COVID-19 patients showed similar results, but these patients were shown to not have a reduced viral load, which is what we would expect if remdesivir was blocking viral replication. So this suggests there could be another mechanism at play with this drug in COVID-19. The full results from this trial have still not been published, so we can't see the viral load of the patients who had this shorter time in hospital. Also, this trial was cut short because the placebo patients were eventually given remdesivir as well, as it appeared to be having a beneficial effect. This was for ethical reasons, but now the study will be much harder to interpret because there aren't proper controls in place. But what we really need to see here is the full results from this trial to get a better idea of how this drug is working. We need larger clinical trials to replicate these results. But we potentially have a drug here that will aid quicker recovery and reduce the burden on the healthcare service. My second science highlight this week is that researchers have been able to get a closer look at the emergency brake for cancer in the body. Cancer is the replication of abnormal cells, and the body doesn't recognise them as foreign cells like it would with a virus or bacterial infection because these cells are your own. Many different mechanisms in our cells can trigger them to become cancerous. Your cell is a hub of activity, and everything is finely tuned and regulated to run smoothly. A cancer cell can be formed where some of these processes in the cell are deregulated. One dysfunctional process which can cause cancer in the cell is the impairment of off-switch proteins which regulate many cellular processes. Of particular interest in this family is an enzyme called PP2A. PP2A activity has been shown to prevent tumour growth. This is because it's able to switch off and therefore regulate processes the cancerous cell needs to manipulate. It's been shown that if you reduce the levels of part of this PP2A enzyme in cells to below half and then insert them into mice, these become cancerous. So therefore, we want PP2A to be on and working so it can act as a brake if a cell starts going down a cancerous route. Scientists can add small molecules to cells which bind to PP2A and stabilise it. And this means that the cancerous processes going on in the cell cannot alter its activity. And research published this week shows the fine detail of this interaction between PP2A and one of these stabilising small molecules. Researchers did this using, again, cryo-electron microscopy to get a really close look at the interaction. Knowing exactly where this small molecule binds to PP2A to keep it on and stable is really important for future drug development, because then we can make drugs which mimic this action but are safe to use in humans. And if such drugs came into existence, they could be used to stop tumour growth. My final research highlight of the week is that we now think the human language can date back over 25 million years. The way we communicate has been key to our evolutionary success, and our language was believed to have evolved about 5 million years ago. But researchers looking at primate brains has found that actually it could have originated about 20 million years before this. Now, I'm not necessarily talking about how we speak today, but more the language structures that we have in our brains. These researchers studied the arcuate facilis in the brain, and this is what we describe as the human language pathway. It connects two areas involved in speech. When this connection is damaged, it can affect the production of speech. This can happen during development or also as a consequence of stroke. These researchers went on to see if there were any other parts of the brain which was connected to this pathway in chimps, humans, and macaque monkeys. These researchers compared MRI scans of human brains and also took MRIs of chimps and macaques. And what they found was that all three primates share part of a pathway which linked the frontal lobe, where the speech and language centres are, to the hearing area of the brain in the auditory cortex. This common auditory segment in all three species implied that we must have had this in a common ancestor. And the last common ancestor of humans and macaque monkeys was over 25 million years ago. So the fact that this connection is in both humans and macaques means it must have developed all that time back in our common relative. It's now time for my Scilight of the week, and this week the Scilight is a little bit different in terms of research style because it is a correlation study, and I don't really rate correlation studies in their isolation, but I think the results in this paper are really important for the dementia research field, so I'm going to talk about them. This paper looks at the link between dementia and inflammation in the brain. What dementia is a syndrome which causes cognitive problems due to the death of brain cells. There are many different diseases which cause dementia, and these different types of dementia can be defined by changes to the brain in specific locations. This paper focuses on frontotemporal dementia, or FTD. This disease can cause a huge spectrum of symptoms. This includes personality changes, impairments to decision making, and also problems with speech and language. In many diseases which cause dementia, a common mechanism found in their brains is that healthy proteins that you normally have in your cells misfold into abnormal shapes. The exact impact that this has on the brain is still uncertain, but it is thought that the misfolding of these specific proteins is linked to the death of brain cells in dementia. A problem that we face is that many different proteins can be found in a misfolded state across the different diseases which cause dementia. But clinically, patients can present with similar symptoms even if they have different misfolded proteins in their brain. You can normally only 100% classify which of these proteins the patient has after they've died. So this makes it really hard to find tailored treatments for these patients. What would be really useful in terms of therapy and treating these patients would be to find a common mechanism which occurs across all dementias, irrespective of what misfolded proteins the patient had in their brain. And one of these potential common mechanisms is neuroinflammation. Your brain has its own immune system, which is vital for keeping it healthy, and one of the main cells involved in this immune system are called microglia. These cells are like scavenger cells, and they clear up debris and other pathogens that may enter the brain. But there is a point, like the immune system in the rest of your body, when too much of this function can become a bad thing. In diseases which cause dementia, defects in neuroinflammation proteins have been shown to give a genetic predisposition to developing some of these conditions. And a recent brain imaging study showed that you can see areas of neuroinflammation in the same brain areas where you see misfolded proteins in Alzheimer's disease. And so the researchers in this study wanted to see if you could see neuroinflammation in the areas of misfolded proteins in the brains of patients with FTD. The researchers looked at 3 classifications of FTD patients, and this is based on their clinical symptoms. In FTD, there are normally 2 proteins found in a misfolded state. These are called TDP43 and tau. So the researchers scanned the brains of 31 individuals with FTD after they've been injected with a dye, which is uptaken in areas of the brain where there are deposits or misfolded aggregates of TDP43 and tau. They also injected these patients with a dye which is uptaken in brain areas which have neuroinflammation, and this is marked by these activated microglia cells. The scans revealed a significant increase in the uptake of this neuroinflammation dye in the frontal and temporal lobes of the FTD patients compared to the healthy control group. And there was also a strong correlation between the uptake of the 2 dyes in specific brain areas. And this indicates that the areas of the brain where these misfolded proteins are also have high levels of neuroinflammation. In addition, and for me, this was the most interesting part of the paper, the uptake of the neuroinflammation dye was able to more readily discriminate the 3 different groups of FTD patients rather than the uptake of the misfolded protein dye. And this means that you could match patients with similar clinical symptoms based on where the neuroinflammation is in their brains. This could mean that neuroinflammation is impacting the specific symptoms of this disease a lot more than we thought. Now, obviously, this is just a correlation study, it's just a link and needs further investigation. But finding a neuroinflammation fingerprint for these different types of FTD could lead to better diagnostics, which then leads to better therapies in the future. Having a fingerprint which can distinguish patients is really useful for grouping them for these future trials and hopefully getting successful therapies from them. And because inflammation was so prevalent in this group of patients and in the Alzheimer's patients from the other paper, we could look at treatments which stop this inflammation happening in the brain, as this represents a potential common target across a lot of the diseases which cause dementia. So overall, a cool paper, like I said, just a link, but I'm really looking forward to seeing all the experimental work that comes out because of these results. Scilight done! This week, my most clickbaity science headline goes to just 10 minutes of sun boosts vitamin D, which might reduce coronavirus risk. I will get to my main gripe with this headline because it is more situational than anything else in a minute. But first and foremost, let's dig in to see the science that was in the article. There was very little. The article talked about the link between sun exposure and production of vitamin D. This is true. And then it goes on to say this could potentially reduce your risk of coronavirus. But where is your evidence? This article has no evidence for a link between vitamin D and coronavirus risk because it doesn't exist yet. Let's talk about this hypothetically. How could high levels of vitamin D reduce your risk of coronavirus? Vitamin D is thought to play a crucial role in your immune system, and you can get it from sunlight, from certain foods you eat, and also through supplementation. In this article, they quote a researcher from Brisbane who said it would make sense if having low levels of vitamin D leaves you more vulnerable to contracting coronavirus, as this link has been shown with other respiratory illnesses. But, and they even quoted this in the article, she said, we need to do more research to establish this link. Previous work has shown that having low vitamin D does increase your risk of contracting a respiratory illness and also having a longer duration of disease. This report looked at 25 studies, which collectively contained the data from 10,000 individuals to see if vitamin D supplementation can protect against respiratory illnesses. The results found that this supplementation was protective, but only really in those individuals who had really, really low vitamin D to start with. The article didn't even cite this paper, so I had to go and find that myself. But despite these results, there is still no link between vitamin D levels and your risk of contracting COVID-19 from SARS-CoV-2. There might be a link there, but you need the data to sort of write an article saying vitamin D may reduce your risk of coronavirus. It's just like, it really annoys me. Okay, but the most annoying thing about this article for me, and if you think that the lack of science wasn't the most annoying thing, this was, it just got me so bad. At the time this article was released, we were in lockdown, and a headline like this makes people want to go outside and lie in the sun. If you have a garden, that's fine. You can go and lie in the sun. If you live in a flat, like I do, and have no outdoor space, people might read this article and go to parks and lie on the grass. And that, at the time when this article was released over a week ago, was not allowed. So I saw that and I got really annoyed, because I was like, you just don't need to fuel some people's want to go and break lockdown rules, and this headline could have done that for some people. So yes, it was bad science and could have encouraged breaking of government guidelines. If you see headlines like this one, a tip from me to check that the conclusion they've made is genuine is to go in and see if they've quoted the original research paper in there. This article was written without even a research paper to back it anyway, but they didn't even quote the research paper which links vitamin D and reduced respiratory illnesses anyway. If the paper isn't mentioned, it's a red flag. Thank you so much for joining me this week. If you're liking the videos, if you're liking the content, subscribe to my channel, and if you have any questions for me or want me to highlight any research for you or anything you see in the news, or just want me to get down with one of these clickbait headlines, then contact me on my social medias. I'm there to answer your questions. I will see you for more science next week. Bye bye.